To successfully reproduce, the human male body solves a complex engineering problem, continuously manufacturing fragile genetic packages, spermatozoa, and safely delivering them into a target environment. Solving this requires a precise, chronological pipeline built on 3 interacting systems. First, there is the raw manufacturing base, responsible for generating millions of gametes every single day. Second, the system requires an internal chemical engine, fueled primarily by the secretion of testosterone, to regulate that production. And third, it needs an active transportation mechanism to physically move those cells out of the body and into the female reproductive tract. This entire system functions as a singular, integrated pipeline of manufacturing, maturation, and delivery. By tracking the physical route of a single gamete from creation to exit, the logic of the anatomy becomes clear. Every structural form in the male reproductive tract is dictated by a strict physiological function. The pipeline begins at the primary sex organs, the paired gonads known as the testes. This cross-section shows the internal architecture. Beneath the dense fibrous capsule called the tunica albuginea, the tissue is divided into distinct lobules, packed with tightly coiled seminiferous tubules. Inside these tubules are spermatogenic cells. This is the factory floor, responsible for the continuous, baseline production of raw sperm. These developing gametes cannot survive on their own. They are flanked by Sertoli cells, which provide the direct structural support and nourishment required to build a viable cell. Just outside the tubule sit the Leydig, or interstitial cells. When triggered by luteinizing hormone from the brain, these cells produce the testosterone that drives the entire manufacturing process. There is a severe biological constraint on this factory. Look at this thermographic map of the male pelvic region. Standard core body temperature is 37 degrees Celsius. At that temperature, spermatogenesis completely fails. The enzymes required to build sperm degrade in core body heat. To solve this thermal constraint, the body houses the testes outside the pelvic cavity, suspending them within the skin-covered sac of the scrotum. The scrotum actively manages its own surface area for heat loss. The dartos muscle sits within the scrotal tissue. When it contracts, it wrinkles the skin to conserve heat, or relaxes to smooth the skin and cool the organs down. Further up in the spermatic cord, the cremaster muscle acts as an active biological elevator. It physically raises the testes closer to the body for warmth or lowers them when ambient temperatures rise. Blood supply is also regulated. A specialized venous network called the pampiniform plexus wraps around the incoming testicular artery. It acts as a counter-current heat exchanger, pulling heat out of the arterial blood so it arrives at the testes pre-cooled. The external gross anatomy of the scrotum, its muscle attachments, and its vascular network serve one exclusive physiological purpose. They create an active, mechanical thermal isolation chamber to guarantee gamete viability. When a sperm cell leaves the seminiferous tubules, it is structurally complete. It features a head, a distinct midpiece, and a long tail, but it is functionally immotile. It cannot swim. To gain motility, it moves into the first ductal stage. This is the epididymis, a highly coiled tube attached to the posterior surface of the testis, divided into the head, the body, and the tail. Sperm are temporarily stored here. As they travel through these coils, they undergo final functional maturation, specifically developing the biochemical ability to move their tails. From the tail of the epididymis, the pipeline turns upward into the vas deferens, or ductus deferens. This is a thick-walled muscular tube connecting the external testicular environment back to the internal pelvic cavity. Because these sperm are not yet swimming freely, they cannot move themselves through this long transit route. Instead, during ejaculation, the thick, smooth muscle of the vas deferens undergoes rapid peristaltic contractions, rhythmically pumping the fluid upward. This muscular tube travels vertically up through the spermatic cord, bundled tightly alongside the testicular artery, veins, and internal nerves. The early transit phase relies entirely on the mechanical, muscular pumping of the duct system. The sperm's own motility is held in reserve. As the raw sperm reach the end of the vas deferens, they lack the energy reserves and chemical protection to survive outside the male body. To solve this, the pipeline integrates a chemical payload. This diagram shows the junction point behind the bladder. The vas deferens meets the duct of the first accessory gland to form the ejaculatory duct. Here, the paired seminal vesicles empty their contents directly into the flow. These glands secrete a dense, fructose-rich fluid. Because sperm carry no stored fuel, this fructose provides the direct, immediate metabolic energy required to power their flagella. The sheer volume of this fructose secretion accounts for approximately 60% of total semen volume. The fluid then drops straight down into the prostate gland, a single organ surrounding the upper portion of the urethra just below the bladder. As the sperm pass through, the prostate squeezes a milky, highly alkaline fluid into the mixture. The environment of the female vaginal tract is highly acidic to prevent infection. Without the prostate's alkaline fluid neutralizing that acid, the sperm would die instantly upon delivery. Finally, just below the prostate, the tiny paired bulbourethral, or Cowper's glands, add a mucus pre-ejaculate. This clears out residual acidic urine left in the internal urethra and provides necessary lubrication. Together, these three accessory glands completely engineer the payload. They transform raw gametes into semen, buffering the fragile cells against the harsh environmental conditions ahead. With the chemical payload assembled, the semen enters the final structural pathway, the urethra. In the male anatomy, this is a shared conduit, acting as a common passage for both urine and reproductive fluids. To navigate the female reproductive tract, the urethra runs through the external genital organ of copulation, the penis. This transverse cross-section shows a specialized architecture of 3 parallel cylinders, 2 dorsal corpora cavernosa and a single ventral corpus spongiosum surrounding the central urethra. The 2 corpora cavernosa are composed of highly expandable vascular erectile tissue. During sexual arousal, these cylinders engorge with arterial blood to provide structural rigidity. The lower cylinder, the corpus spongiosum, performs a different job. It encases the urethra and prevents the tube from collapsing under the intense hydraulic pressure of the surrounding erection. It keeps the duct forced open for ejaculation. At the very end of the line, the corpus spongiosum expands to form the gland's penis, terminating the delivery route. The external genitalia effectively repurposes the urinary exit route, utilizing specialized vascular architecture to force chemically protected gametes into their target environment. Because fertility relies on this linear sequence, any structural anomaly in the anatomy creates a cascading failure in reproduction and often in urination. Take a look at these 3 common clinical obstructions. The first is cryptorchidism, a condition where a testis fails to descend into the scrotum during development. Because the testis remains inside the pelvic cavity, it is continuously subjected to a core body temperature of 37 degrees. This violates the strict thermal constraint required for spermatogenesis, leading to infertility. Moving up the cord, we see varicocele, which is a pathological dilation of the venous network known as the pampiniform plexus. When these veins swell, the specialized countercurrent heat exchange mechanism breaks down. Arterial blood reaches the testis uncooled, abnormally warming the environment and drastically reducing sperm count and motility. And finally, there is benign prostatic hyperplasia, or an enlarged prostate. Because the prostate directly surrounds the shared urethral pipeline, its abnormal outward growth mechanically crushes the central tube inward. This severe stricture impedes both regular urination and the delivery of the ejaculate. The male reproductive tract is built upon a rigid, functional architecture, where every structure serves a specific mechanical or chemical purpose. From the initial division of a stem cell to the final delivery of a chemically fortified ejaculate, survival depends entirely on the unbroken mechanical, thermal, and chemical integrity of this specialized system.